This study that we proposed on, and again, thank you very much to the committee for recognizing uh, our work and for funding this proposal, allowing us to do this important work. Phil Felgner and I have been collaborating for a number of years now, and so I'm a clinician in the Division of Infectious Diseases, and I'm on the KL2 award from ICTS, and Phil is in physiology here. So we've been working together on the platform called the protein microarray, which I'll talk about briefly. So we propose a study to do surveillance for antibodies to COVID-19 during an outbreak. So, we've collected, or our collaborators have collected about 1,000 samples from blood donors during outbreaks in Washington State and New York over the past month. And we propose to use our technology, this coronavirus antigen microarray, to test for antibodies against a panel of antigens, both from SARS-CoV-2 as well as numerous other coronaviruses. And so in order to validate this assay through data that I'll show in just a second, we use convalescent sera from SARS-CoV-2 virus, PCR positive individuals as positive controls, as well as some sera collected prior to the pandemic as negative controls. And we trained a model to basically detect who has been previously infected with SARS-CoV-2. And we will apply this technology to the sera collected during these outbreaks in Washington and New York to try to measure the true disease prevalence over time. Because we know that our PCR-based symptomatic testing strategy is missing a lot of people who are either asymptomatic or minimally symptomatic, but are very important in the spread of the disease. So our antigen microarray, the way this works is we have purified protein antigens that a company that we work with makes and expresses in HEK 293 cells, and they're printed onto these slides. And then human sera is then incubated on these slides, and secondary antibodies are used to detect any human antibodies against the antigens from the viruses. So through this, we generate a heat map that essentially gives us a profile of the person's antibody response against numerous antigens from the virus, in this case, SARS-CoV-2, as well as other coronaviruses to assess for cross-reactivity of these antibodies. So, just to validate that this antibody test actually works, we have in red a set of positives. These are from the patients who are PCR positive and recovered from SARS-CoV-2 infection. And the blue is the negative. And so this is a panel of antigens on the bottom from, on the left in blue, the blue shade is SARS-CoV-2. Interestingly, the light green here is common cold coronaviruses. And then the others are other respiratory viruses. And you can see that the blue bars, which are negatives from previous to the pandemic, don't really have reactivity to the SARS-CoV-2 antigens, whereas the red bars, which are the known positives, do. So this shows that this test is working. And we can also look at the same time at IgA, which is similarly to IgM, is more of a marker of acute respiratory infection, whereas IgG remains positive and is a marker of past infection. And we see the same thing here. We see IgA in some of the convalescent sera. So again, as I mentioned, we're going to apply this technology now to these 1,000 specimens from Washington State and New York and try to figure out, okay, what is the proportion of patients in the community who are asymptomatic or minimally symptomatic here. And this is very important to actually opening up society, and getting us to a point where once we know that around 50% of the population has been infected, that gives us more confidence that, okay, we may be able to open things up. So this study will go a long way towards that. And a couple other studies I just want to mention, the seed funding will start off paying for that initial study, but a couple others that we're working on that you may hear about is we have an IRB to look at serology in healthcare workers at UC Irvine Medical Center at, over the course of the outbreak here, to look at healthcare worker specific risk factors. And then we have another IRB working with the clinical laboratory where they've actually collected clinical remnant specimens from patients who have SARS-CoV-2 infection in the hospital. And we're going to look at some of their clinical factors and look at antibodies and see if certain antibody profiles predict more severe disease or predict people who are more likely to recover. So that's what I have. Thank you. I guess I don't know if we have time for questions, but I'm happy to answer any if we do.